It's a real pleasure to be here and to representing the botanical circle of China and talking about DNA barcoding on the Chinese plants. So talking about plants, I would like to remind you the global strategy for plant conservation, which was adopted in 2002 to halt the loss of plant diversity worldwide by 2010. And so there are 16 targets. The first one actually in five aspects. The first one is plant diversity is well understood, documented, and recognized. And so the first one actually is a working list of all known plants. And so actually this was achieved by a global initiative, including the Kew Gardens, Missouri, New York, and many other botanical gardens across the world. And by synthesize the plant list, including some 300,000 accepted name by that time. But actually, the whole target, basically for 2010, to achieve a significant reduction of the current rate of biodiversity loss at the global, regional, and national levels are not being made. So this is truly a problem, and actually we are a lot of scientists thinking about the Earth is now experiencing the sixth mass extinction by actually something like three quarters or more of the species go extinct in 1 million years time. And so the UN decided to make a decade for international biodiversity now from 2011 to 2020. And the new targets for GSPC was adopted on the 29th October 2010 in Nagoya, Japan. And the first target, again, is an online flora for all non-plants. And so actually, this is a first meeting of the consortium in the Royal Botanical Garden, Edinburgh, in 2013. And so I would like to quickly talk about the plant diversity in China. And everyone knows well about the global diversity in China. It's actually harbors 4 of the 34 internationally important biodiversity hotspots. And China has a very diverse geography from the coast area in the south and the east and to the desert in the northwest. And also, I would like to mention about my own province of Yunnan, which is in the southwestern corner of China, bordering with Tibet in the northwest and west with Myanmar, Laos, and Vietnam on the south. Only 4% of the land territory of China, but harbors 50% of the species of plants. So just a few pictures to illuminate the landscape and the ecosystems, and something like the tropical rainforest in Xishuangbanna, bordering with Laos and Myanmar, and the evergreen broadleaf forest in central part of Yunnan, and the subalpine conifer forest in the northwest Yunnan. Here is in Baimasan of Deqing. And more deciduous and alpine mixed forest in northwest Yunnan. And Yunnan also is a very remarkable province with diverse ethnic people. Actually, 25 ethnic minorities across the province because the high mountain, which the highest is 6,700 meters in with Tibet, and the lowest one is only 74 meters bordering with Vietnam. So this is a high mountains and a lot of isolation for the different people cause the many ethnic groups. So back to the Flora China, the Chinese version was initiated in 1959, and it took 45 years to complete. So the final figure is something like 31,000 species of vascular plants of China, and we quickly moved to the English version, which was in collaboration with the Missouri Botanical Garden, and this one is slightly quicker in 25 years. And the number is almost the same, 31,000, but actually the context is enormously different because a lot of synonym and a lot of new records, new species were discovered by looking at international herbarium specimen and more field work. And so talking about in 264 years, I mean, for whole taxonomists, I mean, we only described something like 2 million species, and the estimation is 10 to 100 million. So we are still a long way to go for even for the plants. And so also because the Flora China Chinese version and the English version was mostly based on specimens in the 1950s and 1970s, so there are a lot of change because of the economic developments. And so the DNA barcoding application, I think now is really a good technique to be applied for barcoding the plants of China. And this is a really well used picture using a standard portion of the genome. And of course, for plants, it's not we cannot use the COX-1, so MAT-K and RBCO were suggested. And actually, this was reviewed, announced in the Mexican conference, and to be reviewed in 1.5 years. And actually, this group in Beijing, Professor Chen Slin's group, was proposing to using ITS2 as a universal barcode for both plants and animals. So, and at nearly the same time, we were lucky to get support from the Chinese Academy of Sciences to organize a national team of something like 50 botanists across 22 organizations to assemble a large amount of data, including a lot of training and standardization of the protocols. And we support, I mean, to use ITS as part of the DNA barcodes because of the larger data set actually indicated that RBCO and MAT-K combination only discriminated on, less than 50% of our data set. But by integrating ITS, the discrimination power can be as much as 70%. So under the umbrella of ABLE, China made a great effort to barcoding its vascular plants. By the last project, we obtained something like 74 million barcodes representing 10,000 individuals of nearly 7,000 species. I think I was giving a talk in Adelaide thinking about 6,000 species, and actually this was achieved by 2014. And now, actually, we will also house what we call a germplasm bank of wild species, mainly focusing on plants and particularly seeds. by last year, we got something like 71,000 accessions of seeds representing more than 9,000, about 9,500 species, which is 32% of the flora. And also we got a lot of DNA samples, and this is really important for our work for barcoding. And this is the seeds using GPS data, mainly from southwestern China, but actually across the country. So the new goal now, we were just lucky enough this year received a new project from the Chinese Academy of Sciences to barcoding more species. In this case, we targeted to barcoding 18,000 species, which is 60% of the vascular plant flora of China in 5 years' time. So this is the target. And now we are proposing an idea called iFlora. Actually, it's maybe a next generation flora by integrating barcodes, plant specimen, diagnostic character, all essential elements of a standard flora, but including DNA barcode and the sequences to be using a iCloud platform, and everyone can use maybe something like a mobile phone to identify their plants. And the key innovation, of course, is DNA barcoding. And of course, using barcoding, we can identify what we call the non-standardized material, including fragmented material, traditional Chinese medicine products, and ecological samplings. And we had a few. I will have you highlight you 2 cases. In this one is a study on Texas, and there will be a talk by my colleague Lian Ming on Thursday, talking about the global DNA barcoding reference lab. So actually, this was a disaster for these plants because of the discovery of Taxol as an anti-cancer property. And so we spend a lot of effort to understand the crypto species, including the OME type now, and a species used to be called Florinia, and we actually used to be called 2 to 4 species, but actually it's 7 in China. And we also managed to identify something like this from the local public security bureau to be a species actually which is used to be called Hendan type. Actually, it's Textflorinia. And but the problem for it's not always successful for the first generation barcoding. In this case, bamboo, and it's really amazing. And actually, I studied this group plants for more than 30 years, and lots of different genera, different morphological character, and in some of them, I mean, do produce fruits like this, like a pear actually, and we made a lot of effort using DNA sequence data to understand the phylogeny and to distribute the 2 major subtribes. And talking about the template clade, which is mostly in Eastern Asia, only a few species in Africa and Madagascar, and 3 species in North America. And we used a large number of the chloroplast sequence, only distributed 10 major clades, but some of these clades, with the biggest one is including 19 genera in the traditional taxonomy. So we tried to use the conventional barcoding to separate the bamboo, but actually this is really, really disparate. Only less than 15% of the species can be distinguished. And so we tried to sequence the whole chloroplast genome And eventually, we using the whole crop genome, we can get some much better resolved phylogenetic tree, including some of the very long internode and some of the very, very short internode. So this is something, maybe a radiation across this area, and we later dated it to be something like 7 to 8 million years ago. And actually, by looking at the chloroplast genome, and we are thinking about DNA Barcoding 2.0, and papers like this one in American Journal of Botany in 2012 talking about using the whole chloroplast genome and liposomal DNA sequences, and actually, we also look at a genus called Symphedium, which is a very important ornamental orchid in China, and we being able to distinguish the individuals, different cultivars, by using the whole chloroplast genome. And another example is Camellia, which is also very difficult genus because of the polyploidization and hybridization. So we also able to distinguish some of the very closely related species. And of course, plastic genome is widely used in projects like Fellow Alps and in a lot of the fellow genomic studies. And later on, genome skimming becoming very useful, and a lot of paper like this one in American Journal of Botany in 2012 and more in 2014. And we also developed a long PCR primers to sequence the plastic genomes. And by now, we are actually assembled something like 2,881 plastons all across the APG tree. And this is something we are still analyzing at the moment. And of course, we worked on things like the Losisi, including very difficult clade of the mallows and the pears and the apples. Actually, and we managed to look at find that the differentiation is in the late Cretaceous, particularly after the KT boundary. And we also look at using chloroplast genome. We also managed to look at the T family, which is a lot of genus are evergreen species in the broadleaf forest, and we managed to find that during the Oligocene and the Miocene boundary, and there's a shift of diversification rate, and this may be triggered by the Asian monsoon system. So right now we assembled some 4,000 species of plasmon of vesicular plants for China, and the target, as I mentioned, is 18,000 in 5 years' time, representing 80% of the general and all families of China, and maybe 98% of the family of the APGs, because we got all the Hebelian specimen and the seed banks and the pipelines available. So this is going to be the target. And finally, I would like to mention that we also made an attempt to genome skimming Hebelian specimens. In this case, we got 25 specimens. The oldest is 80 years old, including TAC, RAC, Lolaceae, Margoniaceae, all different families. And we actually assembled 22 of them with whole chloroplast genome and 24 with rDNAs. And this starter input DNA is as low as 500 pigs. And so there will be a talk this afternoon giving you more details about this technique. And so I think as botanist, I mean the using of Hebelian specimen is a milestone for taxonomy. And of course, I mean Linus, I mean the use of binoning for plants is also another one of the most important steps. And also, I think the barcode will be the startup of digitalization of genetic information of species. And so it is now the time to integrating all the information, including Hebelian specimen, including the GPS data, make everything more accurate, repeatable, predictable, and faster. So to conclude, I think if we are doing inventory, a checklist will be the starting point. But if a virtual checklist is much better because you can trace it, you can, going back to the Hebelian to look at the specimen. But if you got the checklist also tagged with a digitalized DNA barcode or DNA sequences, I think there will be really much more information, much better than a single Hebelian specimen, particularly the older specimens. So I think the botanists should and can do a better job in integrating DNA sequence data with Hebelian information, and of course functional traits, geological information system, internet, and computer technologies. And so finally, I would like to thank the founders, my collaborators, and organizations in supporting this work. And thank you all for your listening.
All right, thank you, De-Zhu. Do we have any questions for him? Going.
It's a wonderful talk. And nowadays, people try to use the corpus genomes as a sort of identification tools. How much edge can one get compared to the individual barcodes? We know that we also use several markers in the corpus genomes as a barcode, which seems to be cheaper and easier to attain.
Yeah. Actually, I think chloroplast it's something like the version 2 of the barcodes, because the first version, of course, is RBCL, maybe plus ITS, but this can only discriminate something like maximum, I think, is 70%. But using the whole chloroplast genome, in most cases, we can get something like 80 or plus. But actually, as mentioned by my colleague Peter Hollingsworth yesterday in his talk, maybe we should also think about more about nuclear data, which I think will be the version 3 for barcodes. But at the moment, I think because chloroplast is so important for plants, I think it is really important to get a reference sequences for, at least for the general, and if possible for the species as the second step. And for the really closely related species, like my study on the bamboos, we still struggled by using chloroplast to describing very closely related species. In that case, we actually used nuclear data to reveal the species.
All right. Any other questions? All right. there's one there.
Thank you very much. It's just so impressive, just the number of species you have and what you're able to do, and things are running. It would actually be really great if we other smaller groups could take some advantage, because you're doing high throughput of everything. And I think there's a lot of things, just how you process your species, how you assemble them, and well, what you have tested in terms of taxonomic resolution of the different organellas, and a lot of things that we really would love to learn from you.
Thank you for the comments. Actually, we were lucky because we have a large team. I mean, from collection to lab work to data analysis. But really, this is a joint effort from many groups of botanical organizations in China. I mean, Skimming is, of course, play a central role, but we also working closely together with our colleagues in Guangzhou, in Beijing, in various parts of the country. So actually, this is an enormous job. Think about the Chinese version, 45 years, the English version for 25 years. So I think if we can do it in 5 to 8 years, this will be really an important job for the Chinese flora. Thank you.
Thank you, De-Zhu. Another round of applause.